name is Riley Green. This PowerPoint is a brief overview of cloning and talks about the key points from my Rogerian essay. It highlights the key reasons as to why people are for and against cloning. Everyone has thought about what it would be like if he or she had a clone. There have been many debates on whether cloning should be illegal or not, especially human cloning, which brings us to the question, what is it? A clone is a precise genetic copy of an organism. Although human cloning has not been accomplished yet, animal cloning has already been accomplished. The first successfully cloned animal was a sheep named Dolly in 1996 at the Roslin Institute in Scotland. She lived for about 6.5 years before she died of a type of lung infection that is common in sheep. More animal cloning has been done since then, and it is now possible to clone a pet. But before we get into that, there are two types of cloning, which are reproductive and therapeutic cloning. First, let's talk about reproductive cloning. Dolly the sheep was cloned using this process. The process of reproductive cloning is called somatic cell nuclear transfer, or SCNT, and it has 3 steps. First, remove the nucleus of an unfertilized egg. Next, insert the nucleus of a cell from an animal that is to be cloned into the egg. Finally, place the new embryo into the uterus of a surrogate mother. The next type of cloning is therapeutic cloning. Therapeutic cloning has almost the same process as reproductive cloning, but the embryo is left in a laboratory for several days to undergo cell division until it becomes a blastocyst. These blastocysts can then be used to become any type of cell. People are generally more in favor of therapeutic cloning rather than reproductive cloning. Cloning has many more benefits to people, especially medically, but many others do not believe in it. The major reasons why people are against cloning is because it raises multiple moral and ethical questions. Can the cloning technique harm the clone by damaging its DNA or by condemning the clone to premature aging? Is life destroyed by cloning? For example, therapeutic cloning produces stem cells and takes them from a blastocyst for cloning, which, after a certain amount of days, could have the potential to become a living thing. With this in mind, many people think that by removing the stem cells from it, scientists are taking away a life. Many religious people are also against cloning because they believe that scientists are trying to play God. Although there are many negatives when it comes to cloning, there are just as many positives, and those positives have benefited people in many different ways. Cloning animal models for diseases could help scientists study one form of a disease with a large number of identically cloned animals so that many other animals do not have to die. Cloning also has the potential to revive endangered and extinct species, but scientists would need a very well-preserved sample of DNA and a surrogate mother closely related to the species in order to do this. Cloning has also been able to help people reproduce a deceased pet. The domestic animal to be cloned was a cat named Cece on December 22, 2001. She did not look like the cat she was cloned after, named Rainbow, because certain parts of her chromosomes turned off. This process is called X inactivation, which happens normally in females in order to prevent them from having twice as much X chromosome activity as males. It also happens randomly, meaning that different cells turn off different X chromosomes. A few other benefits is that cloning can also help solve many issues in the medical world. For example, scientists could clone a cow or other animals such as goats and sheep for drug production, and the drug would be in its milk so that when it reproduces, the cow's offspring would have the same drug in them. It could also help for people that have 2nd or 3rd degree burns, because they can have their cells cloned and eventually used to replace the dead skin cells of the burn mark where their skin regenerates itself and almost completely heals. The process of cloning is still evolving, and maybe someday soon scientists will be able to clone a human. There are many positive and negative aspects of the different types of cloning. For example, reproductive cloning can help with drug production and disease models, but it can cause problems with the clone. People mostly fear that human cloning is possible or that it will be possible in the future. Therapeutic cloning can help treat serious scars or burns, and it has the potential to cure other diseases, such as brain diseases. But it does also have the potential to cause problems medically, like giving a person cancer. It has been almost 25 years since the first successful cloning of an animal, and there have been many more successfully cloned animals since then. Even though people find the process of cloning morally wrong in many aspects, others believe it can be used for medical purposes. The possibility of cloning humans may be achieved in the distant future, but for now, cloning advances should be used to potentially cure certain diseases. Thank you for taking time to watch my presentation.